Okay, so in this lecture, we're going to discuss osmoregulation, which is the process of maintaining the levels of solutes in the cells of the body within a specific, relatively narrow range, which of course is necessary for maintaining the proper conditions for normal physiological processes and chemical reactions to occur. So we're going to discuss a few definitions here in a moment, but it's important to recognize that commonly the level of solutes in the body often differs considerably from the concentration of solutes in the surrounding water that fish inhabit. Because there are different levels, different absolute concentrations of solutes, and different levels of individual molecules in the body relative to the environment, active processes are needed to combat the processes of diffusion and ion loss. So if freshwater fish are going to maintain concentrations of solutes higher than the environment, if saltwater fish are going to maintain concentrations of solutes lower than their environment, they're going to need to use some active processes. And by active, I mean they're going to have physiological and morphological adaptations, but they're also going to need to expend energy to support these processes. So depending upon the conditions, there may be a need to maintain levels of certain ions and eliminate others. And that applies to both freshwater and saltwater conditions. So just a few definitions so that we're all starting from the same point. So osmoregulation is the process of maintaining those levels of solutes in the body that are different from the levels of the same solutes outside the body. So then we've got hyper, hypo, and isotonic solutions. So when you see tonic, that's generally referring to solutions, not to cells or biological organisms. So this hyper means to have higher concentrations compared to something else. Hypo means lower, and iso means equal. So if we say that this solution is, if solution A is hypotonic relative to solution B, that means that the concentration of solutes in solution A is lower than in solution B, and vice versa. So if we talk about hyper, hypo, and isoosmotic, we're saying basically the same thing, only now we are talking about cells or organisms. So these, of course, mean that the concentrations of solutes in the organism or in the cell are higher, lower, or equal relative to their surrounding environment. Okay. So there's a couple of different types of approaches that organisms can take. They can be osmoconformers, which means that they maintain body fluids at an equivalent osmolality relative to their environment. That's the conforming part of it. So they are conforming the total osmolality of their tissues to the environment. So what exactly does osmolality mean? Well, it's kind of a combination of terms. Osmo, referring to concentrations of solutes, that sort of thing, and molality. So I'm sure from your basic chemistry class you remember molarity versus molarity versus molality. So molarity, of course, is a measure of concentrations of solutes, but it's expressed in terms of units of the solute, like moles, per liter. So it's per unit volume of whatever the solvent is. So if we're talking about a solution of salt in water in molarity, it would be in moles of sodium chloride per liter of water. If we were talking about molality, it's the same kind of unit in a sense. It's a measure of concentration, but it's expressed in terms of the mass of the solvent. So instead of moles per liter of solution, like you would see with molarity, it would instead be in terms of moles per kilogram of water. So just slightly different units, liters versus kilograms of water. Generally speaking, osmolality and osmotic concentrations are expressed in molal terms. So units of mass or moles per kilogram of plasma or body fluids. Okay, so again, going back to the idea of osmoconformers, they maintain their body fluids at an equivalent osmolality. So the concentration, the sum total of all of the different solutes in their body is relatively equivalent to the same concentrations of all the other different solutes in the water. However, what's important to recognize is that this does not mean that the composition of those solutes is the same. It's most certainly not. So in the body of the fish, concentrations of sodium, chloride, phosphates, bicarbonates, ammonia, amino acids, fatty acids, glucose, hormones, all these different molecules contribute to the overall osmolality. The total concentration of stuff in the body of the fish might be the same as the total concentration of stuff in the environment, but the different components of that that make the different molecules that comprise the total amount of stuff are going to be very different. You can think, of course, the concentrations of glucose are going to be much, much higher in the plasma of a fish than they would ever be in the environment. So that's one strategy, is to be an osmoconformer, to match the total concentration of solutes in your body to the environment. The other strategy, which most fishes adopt, is to be an osmoregulator, which means that you are maintaining your body fluids at an osmolality that differs from the external environment. That could be higher or lower relative to the environment. So you could be a hypoosmotic or a hyperosmotic organism relative to your environment. But regardless, for all osmoregulators, whenever the external osmolality changes, generally speaking, the internal osmolality remains the same, as long as the fish is able to maintain its osmoregulatory function. So osmoregulation is based upon, again, exchange and movement of materials and controlling those processes. So the processes that we're talking about here depend upon the compounds that we're talking about, or the molecules. So water, ammonia, urea, and other small non-electrolytes, in other words, non-charged, things that aren't ions, can move through defects or spaces in the cellular membranes relatively easily. So as you'll see, there are gaps between the cells that comprise the epithelial surface of the gill, for example, and these small non-electrolytes commonly move through them with relative ease. So that can be somewhat challenging to control. Now, electrolytes, on the other hand, so anions and cations, generally speaking, have to move through transmembrane transporters, like sodium-potassium pumps, or other transporters that facilitate their movement through the cell membrane. So because they tend to be larger, and or because they have this ionic charge, it makes it more difficult for them to just move in between spaces or defects, and so usually they need those transporters for a little bit of help. We'll talk a little bit more about that. Just to keep in mind, there are various tissues that or organs that are involved in osmoregulatory processes. The kidney is the primary organ that's involved in osmoregulation for fish as well as for higher vertebrates. Gills are a secondary organ that are involved in osmoregulation. So although we talk about the gills to a large extent in this lecture, it's important to understand that in terms of the number of ions or solutes that are being exchanged, the kidney actually processes a lot more than the gills do in most fishes. But again, those two are probably neck and neck, so to speak, in terms of competition for how much they are involved in osmoregulation. So the kidney and the gills are certainly the top, but there are other tissues that are involved. The gut, the intestine, is certainly involved in osmoregulation. The integument, or the skin, is involved in osmoregulation. Generally speaking, it's involved in preventing the loss or exchange of materials. The urinary bladder, of course, is being a portion of the kidney and the excretory system. It actually has some functions as well. And for those species that have them, specifically the elasmobranchs, something called the rectal gland is also an important contributor to osmoregulation. Now, as we discussed previously when we were talking about the circulatory system, the secondary circulatory system may also be involved. It has close proximity to various epithelial or skin tissues. It has a lot of the body fluid volume contained within it. We know that it's not involved directly in gas exchange or respiration activities. So keep in mind that that secondary circulatory system may actually be a pretty strong contributor to osmoregulation as well. So let's take a look graphically at what some of these strategies might look like. So on the x-axis you have environmental osmolality, so we're going from pure fresh water on the left-hand side to full-strength seawater on the right-hand side, and then you can see on the y-axis we have the fish osmolality. So if we were talking about an osmoconforming fish, As the environmental concentration of solutes, or salinity essentially, increases, the osmolality of the fish will also increase. That's the conforming part of osmoconformer. Of course, osmoregulators maintain their osmolality at a set point that's appropriate for their physiological processes, regardless of what's going on in the environment. So this is generally speaking what these two strategies look like, but it's important that there are limits to these strategies. So here we're showing a little bit more realistic graphical depiction of what these strategies might look like. So if we're looking at the white curve actually represents what's referred to as a euryhaline fish. So euryhaline is from the Greek for euri, meaning wide or broad, and haline referring to salt or salinity. Okay, so a euryhaline fish can tolerate a wide range of environmental salinities. So these might be species like striped bass, for example, that live in the open ocean, but also commonly spend a lot of time in estuaries. They migrate in and out of freshwater to spawn, etc. So they're able to tolerate a wide range of salinities. And that's what's indicated by the relatively broad flat line in the middle. So over a wide range of environmental osmolalities, the fish is able to maintain their osmolality at a set level. However, there are limits. If you put a striped bass in water from the Dead Sea that is much, much more highly saline, at a certain point they won't be able to cope with that. And so outside of that upper threshold of their salinity tolerance, you can see that they begin to osmoconform. And ultimately, their physiological processes would break down and mortality would occur as a result of that. You see the same thing on the lower end of the spectrum. Now, this isn't to scale, naturally, but if you have a euryhaline fish and you transfer it to water that is outside of its salinity range, you can also see it start to osmoconform on the low end of the spectrum. What makes a euryhaline fish is that the range in which it's able to maintain its osmolality is very broad. So it is able to regulate levels of solutes in its system over a broad range of environmental osmolalities. Now, the other two lines that you see here are the opposite. These are stenohaline fish. It's again from the Greek for stenos, meaning narrowness. So these are fish that occupy a narrow range of salinities. And you can have either freshwater or saltwater stenohaline fish. So in the case of the pink line here, this represents a freshwater stenohaline fish. So you can see that they also have a flat horizontal component to that curve, and it's on the lower end of the environmental osmolalities. Okay, so during or when the fish is exposed to normal freshwater, it's able to maintain its osmolalities at that set point, which is appropriate for that fish. But if you drop below that, or certainly if you go above that, the fish begins to osmoconform, indicating that its physiological processes and osmoregulatory function are breaking down, and ultimately mortality is going to occur. What makes this fish stenohaline is that the range under which it's able to maintain osmolality and to effectively osmoregulate is quite narrow. It just happens to be on the freshwater end of things. Now, the purple or bluish line here represents a marine stenohaline fish. So you can see again that it has a relatively narrow range under which it's able to osmoregulate and maintain its osmolality despite what's going on in the environment. And above that, and certainly below that, it osmoconforms, indicating osmoregulatory failure. Okay. Okay, so let's talk about osmoregulation in a teleost in freshwater. So first of all, the gradients are relatively small. So the osmolality on the fish is typically about 300 milliosmoles per kilogram. That's the typical unit for osmolality in the tissues and blood of fish. So approximately 300 milliosmoles per kilogram, as opposed to less than 5 milliosmoles per kilogram. So the total range here is less than 300 milliosmoles, so not as much of a concentration gradient generally. However, there is still a concentration gradient, and so the higher concentration of solutes in the fish is going to draw excess water. As a result of this, the fish does not engage in any drinking behavior like we'll see in some of the other saltwater fishes. And to eliminate this excess water that does diffuse across the epithelial tissues, the fish compensates for this by producing a large amount, what's often referred to in the textbooks as copious amounts of dilute urine. Now, some salts are going to be lost via the urine and in the feces, but they're going to be constantly absorbing ions to replace those that are lost through the means of what are called mitochondrial-rich cells. They're also referred to as freshwater adapted chloride cells. So that's going to be a primary source of ions coming into the body. This is how they maintain those high levels of ions, primarily by importing them to the gill. But of course, they are also going to absorb some of those ions from the food that they consume. Now, in the case of a teleost osmoregulator in saltwater, the gradients here are relatively large. So you can see that the osmolality, typical osmolality of a marine teleost is about 400 milliosmoles per kilogram, so somewhat elevated to the 300 that we saw in freshwater teleosts, but it's not substantially higher than what you see in freshwater. Conversely, in the case of the environment, that is substantially higher here. Of course, we're talking about salt water. So the water is approximately 1,000 milliosmoles per kilogram. So whereas the gradient was less than 300 in the freshwater fish, here the gradients can be 600 to even higher milliosmoles per kilogram. So the gradients are much greater. This results in more energy needing to be expended actually to compensate and to maintain solutes in the body, water levels in the body relative to the environment. So of course in this case the fish is hypoosmotic to its environment, and so the environment is going to draw water from the body of the fish. So to compensate for this, it engages in very high drinking rates. So it's continuously drinking. For example, there is a species called the mummy chug, Fungulus heteroclitus. It's a euryhaline species, actually, that's found along the Atlantic coast. So it's, depending upon the time of year and where this particular individual is at, it may be in freshwater or it may be in full-strength seawater. When those individuals are in full-strength seawater, drinking increases. The amount of water that's brought in is 3 to 10 times higher compared to when it's in freshwater. So they actively drink and consume water. Now, of course, when they consume that water, they bring it in and drink it, they're going to be bringing in a lot of salts as well. And so they have to compensate for that. Now, the urine flow in these species is relatively low, so they don't produce a lot of urine. It's not a principal means of excreting salts, although they do produce some salts and excrete them via that scant urine. They're also going to lose and excrete some salts via the feces, but the principal location of salt export is going to be at the chloride cells in the gill. So before we saw with a freshwater fish, we have freshwater adapted chloride cells, also referred to as mitochondrial rich cells. Those are going to be actively importing salts like sodium and chloride into the gill and into the body in freshwater fish. In the case of marine osmoregulators, they have saltwater adapted chloride cells which actively export those two ions, sodium and chloride. Now, with respect to diadromous fishes, which are going to be in different parts of their life in either freshwater or saltwater, their physiology and their osmoregulatory process is going to vary based on whether they're in freshwater or in saltwater. So here we're showing the conditions for a lamprey in freshwater. So once again, you can see that the gradients are relatively small. So the serum here is shown as being approximately 225 milliosmoles per kilogram. And again, keep in mind that the freshwater osmolality is commonly around 5 or less. So relatively small gradient, but the body is still going to draw water. To compensate for the loss of ions, there's going to be active uptake of both sodium and chloride at the gill, as well as from the food that they consume. And the filtration rate, what's referred to as the glomerular filtration rate, or GFR, is greatly increased in the kidney. So this increased filtration rate allows for the kidney to retain more of the ions that are in the bloodstream and excrete more of the water that's there. So you can see that term there, copious amounts of dilute urine being released. Okay, so this is under the freshwater scenario. Now, the same fish under saltwater conditions, again, they're going to be facing greater gradients. You can see that the serum or the plasma of the fish becomes slightly elevated. So before it was about 225, now it's about 265. So they're flexing a little bit here, allowing their osmolality to increase slightly. It's still within the range of what is appropriate for the lamprey physiologically, but by allowing that to conform a little bit to the environment, they're making their job a little bit easier in terms of balancing and osmoregulating. So it's a little bit higher, it's 265, but that's still quite low relative to the environment, which is again roughly about 1,000 milliosmoles per kilogram in salt water. And so as a result of that, the fish begins to engage in seawater drinking behaviors. Now their gill has become adapted. The freshwater adapted chloride cells have been replaced by saltwater adapted chloride cells, so they are now actively exporting both sodium and chloride at the gill. Now in this case, their kidney GFR, or glomerular filtration rate, is reduced. They don't care if they keep those extra ions. In fact, they'd rather be rid of them. So the urine is now scanty, so they don't produce much of it, and it's relatively concentrated. This is also a primary means by which they excrete some of those divalent ions. So monovalent ions, so they have a +1 or a -1 charge. That's mostly sodium and chloride, mostly being excreted at the gill. In the case of the divalent or +2 or -2 ions, those are going to be excreted primarily in the urine. So those have been some osmoregulatory strategies in freshwater, saltwater, or a species that engages or utilizes both of those types of habitats. But what about the osmoconformers? First of all, you only see marine osmoconformers. You only see osmoconformers in the marine environment. If a species were trying to osmoconform in freshwater, the concentration of solutes that would be tolerable in their body would be close to 0. Obviously, that's not going to be physiologically appropriate. They need to be able to have salts and amino acids, fatty acids, glucose, hormones, all these different things, all these different solutes in their body. And so it's not reasonable to be able to achieve basic physiological processes or engage in normal functioning at those very low concentrations. So you never see osmoconformers in a freshwater environment, but you do see them in some marine fishes, specifically in the elasmobranchs. Now in this case, the gradients that they face are relatively negligible. So you can see that the water here, once again, is about 1,000 milliosmoles per kilogram, whereas the fish itself has concentrations of total solutes approximately 1,100 milliosmoles per kilogram. The way the fish are able to achieve these very high concentrations of solutes in their bloodstream and in their tissues and fluids is that they retain urea. So urea is a byproduct of nitrogenous waste excretion. Those of you who have taken some of my other classes, like aquaculture, we talk about ammonia excretion at the gill. That is what most fishes engage in. But for higher vertebrates that can't directly excrete ammonia continuously, that have to essentially urinate, those species primarily retain or accumulate nitrogenous waste, not in the form of urea, but in the form they don't produce it in the form of ammonia, they retain it in the form of urea. This is because urea is slightly less toxic than ammonia. So if you're an organism like a freshwater or saltwater teleost, you can continuously excrete ammonia at the gill and eliminate this very toxic nitrogenous waste. If you are a terrestrial animal that can't be continuously urinating, you can't release directly, and so that nitrogenous waste is going to build up in your bloodstream in one form or another. If it builds up in the form of ammonia, it's going to be very, very toxic and poison the animal very, very quickly. If, however, they convert that ammonia into urea, which is less toxic, they can accumulate it to the point where the organism can eventually find a location that's appropriate to urinate and eliminate the waste that way. So the marine osmoconformers are a little bit different in that they could theoretically excrete ammonia at the gill quite easily, but they instead choose to accumulate ammonia, convert it to urea, again, to take advantage of the slightly lower toxicity of urea, and use that as an osmolyte. So they accumulate this urea, but again, urea is less toxic than ammonia, but still ultimately toxic. And at the concentrations that these fish accumulate urea, it would normally result in mortality. But they also retain something called TMAO, trimethylamine oxide. This is a compound which essentially serves to counteract the toxicity of the urea. So they're accumulating a lot of urea. They know that that's going to create some toxicity issues, so they produce and retain TMAO to counter those effects. Okay, so again, both urea and TMAO are going to be retained by the kidney and the gills to maintain that high osmolality. Now, the body is going to be drawing water at a slow rate, so they are going to release some water via the urine. They're also going to eliminate excess divalent ions via the urine. They'll also eliminate some additional salts via the feces. But keep in mind that although their total concentration of solutes is matching and actually slightly higher than what's in their environment, they are still going to face a continuous influx of both sodium and chloride. Again, keep in mind that this is salt water. The primary contributors to the salt in salt water is sodium and chloride. So those are much higher in the environment than they are in the fish. And so the fish is going to be continuously facing this influx of sodium and chloride that they don't want. And so to eliminate that excess sodium and chloride, these species have what's called a rectal gland. The rectal gland is where you might imagine it would be. Its purpose is to excrete sodium directly. And in the case of these species, they also have chloride cells, which facilitate chloride excretion. So although they're not facing the same gradients, the same osmoregulatory challenges as most marine fishes, the marine osmoconformers do have to regulate concentrations of specific ions. And so in this case, they're adapted to eliminate the excess sodium via the rectal gland and the excess chloride via the chloride cells in the gill. The cells that are actually in the rectal gland are very similar to the chloride cells that are present in the gill. So very similar function in both cases, just two different ways to eliminate excess sodium chloride. Okay, so let's get a little bit more detailed here in our description of how these chloride cells work. As I'm sure you have assumed, the chloride cells are really critical in terms of osmoregulatory function. So let's see how they actually work. So first we're going to begin with a chloride cell adapted to seawater conditions. So in this case, the purpose of the seawater adapted chloride cells is to export excess sodium and chloride. Okay, so these are going to be higher in the environment than they are in the fish, so the fish will have to continuously export those ions to maintain their proper osmolality. Okay, so this is the primary function of the chloride cell is to be an extrusion mechanism for that elimination of excess sodium and chloride. And the way that the cells actually achieve this is through active transport of chloride. In other words, they're expending energy to export the chloride ions, but then they are passively exporting the sodium. And we'll see how that happens here in just a second. So first, let me orient you here in terms of the structural elements of the chloride cells. So you can actually see the chloride cells, the large cell here in the middle of the slide, and you can see it's supported on either side by two different types of cells. You can see that there's an accessory cell on the top and a pavement cell on the bottom. These are, again, just modified epithelial cells that are found in the gill. The difference between a pavement cell and an accessory cell is basically how tight the borders are between the pavement cells, accessory cells, and the chloride cells that they support. So you can see on the bottom, you can see the pathway or the space between the chloride cell and the pavement cell is labeled as a tight junction. What this means is that it's very difficult for osmolytes to pass through this particular pathway or this defect, this gap essentially between the cells. The gap is relatively tight, and it's actually much longer than this diagram would imply. So if you think about this junction between the two cells as a pathway or a road that osmolytes could travel down to get from the blood on the right-hand side to the seawater on the left-hand side, in the case of the pathway between pavement cells and chloride cells, it's a very long, winding road. And so it takes a long time for osmolytes to travel that path, and typically they're grabbed up and absorbed by the chloride cell along that path before they have a chance to fully exit and get into the seawater or get into the blood. So these tight junctions are relatively impermeable, and osmolytes can't pass back and forth between them. Now, on the other hand, at the upper portion of this figure, you can see the accessory cell and the chloride cell have a relatively, what's called a leaky tight junction. So it's still a relatively tight junction between these two cells, but what makes it leaky is that it's not as long or circuitous a path for osmolytes to travel to get from blood to seawater or from seawater to blood. And so it's leaky with respect to these osmolytes, so they can move in and out of this with relative ease. Okay, so how does this actually work? Well, when you're looking at these figures, or really any kinds of diagrams that show active transport, I recommend that you follow the money, or in the case of biochemistry, follow the ATP. ATP is the energy currency of cells. That's where the money is. Okay, so you can see on the right-hand side here we have a cotransporter that's labeled with ATP. So that means that this is an ATP-dependent pump. So this is transferring sodium and potassium back and forth between the chloride cell and the blood. And so this, utilizing ATP, expending energy, this pump or this transporter exports 3 sodium ions into the blood from the chloride cell, and then does so in exchange for 2 potassium ions coming in. Okay, so what this does is it creates a high positive charge and a high sodium concentration on the blood side. Remember, it's exporting 3 positive ions into the blood, but it's only taking 2 positive ions in. So that's creating an abundance of positively charged ions on the blood side. And again, it's also creating a very high concentration of sodium in the blood. So the sodium is going to want to diffuse down its concentration gradient and also down its charge gradient back into the chloride cell. But the way the chloride cells are adapted is that they can only come back into the chloride cell through the second transporter. So you can see the second transporter there towards the bottom of the chloride cell. Now this doesn't have ATP on it. This is a passive transporter. But basically it only allows the sodium to come back into the chloride cell if it brings some friends with it. Okay, so it has to bring at least 1 potassium ion with it and 2 chloride ions. This is the only way that this transporter works. So although that's high concentration of sodium that's being built in the blood, all the sodiums really want to go down their concentration gradient and get into the chloride cell. They can only do so via this type of transporter. So they're going to have to bring those friends with them. I often joke that the chloride cell is like the nightclub, the transporter is like the bouncer, and the sodium is like a bunch of guys that want to go dance. The only way that all these guys are getting into the club is if they bring some girls with them. So the only way that sodium is getting in is if the 2 chloride cells and the potassium ion come along with them. Okay, so we're expending energy at one point. We're utilizing ATP to initiate this process to create this artificial positive charge and high sodium concentration in the blood, but that in turn is going to drive the export of chloride as well as potassium from the blood into the chloride cell. So ultimately, as a result of that, the chloride ion concentration is going to build in the chloride cell over and over and over again as this active and then passive transporter as these two work together. Okay, so the 2 chlorides are coming in, being driven by and dragged in by the sodium ions, in spite of the fact that the chloride is being imported against its concentration gradient. So concentration gradients or chloride concentrations build within the chloride cell to the point where they're actually higher than what you have in the environment. So ultimately, the chloride is able to passively diffuse down its concentration gradient into the seawater. And again, it's only able to do so if it achieves a very high concentration gradient. Okay. Meanwhile, this sodium is still continuing to build within the blood. That ATP-driven transporter, that sodium-potassium pump, is continuing to drive the sodium concentrations higher and higher and higher. Again, the sodium in the blood is going to want to get away from the blood. It's going to want to go down its concentration gradient. We've already talked about the one option. It can go into the chloride cell again if it brings a potassium and 2 chlorides with it. That's one option. The other option is for it to work its way down through the leaky tight junctions. And so that's what you see here. You see that the sodium is building to the point in the blood where its concentration is so high that it's going to be forced down that leaky tight junction. That's the only other option. It can get into the chloride cell via the passive transporter as long as it has potassium chloride to come with it, or it can go down the leaky tight junction. And again, it's going to build to a concentration that's higher in the blood than is in the seawater, and it will ultimately passively diffuse out through that junction, or what's called a paracellular pathway. So this pathway, paracellular, alongside the two cells. This is just another depiction of how this works in a lamprey found in salt water. So you can see we have the accessory cell, AC, we have the mitochondrial rich cell, or the chloride cell, the larger one, and you can see that we have our potassium sodium pump that's actively using ATP to pump out 3 sodium ions back into the blood in exchange for 2 potassium ions coming in. We can then see the sodium trying to diffuse back down its concentration gradient into the chloride cell through the transporter on the right. So it has to bring a potassium and 2 chlorides with it. It's going to be bringing that along with. And eventually those chloride concentrations are going to build to the point where they are able to exit and diffuse through a passive transporter at the surface of the chloride cell and move into the salt water. You can also see the sodium passively diffusing down that paracellular pathway, or the leaky tight junction. So keep in mind it's a fairly efficient process. We're only utilizing ATP at one of these places, at that initiating sodium-potassium pump. The rest of the processes are active, but again, everything depends upon the functioning of that sodium-potassium pump. So as long as that thing continues to operate and utilizes ATP to do so, the rest of these processes will happen passively, not utilizing any energy. Okay, so that was functioning of the chloride cell in a saltwater situation. Let's take a look at how freshwater adapted chloride cells work. So you can see a very similar arrangement here, except that the first thing you should notice is that there are no accessory cells. So the chloride cells in freshwater adapted gills are surrounded by pavement cells on all sides. And these pavement cells, again, are the ones that have the tight junctions. So unlike the saltwater adapted chloride cell, the freshwater adapted chloride cells have no leaky tight junctions or leaky paracellular pathways. They're all tight all the way around. The structural elements, like the saltwater adapted cells, these have many mitochondria. This is to produce the needed ATP to drive the ion exchange processes. Now, one thing I should have mentioned on the saltwater side, structurally on the surface of a saltwater adapted chloride cell, there's what's called an apical pit. We have a figure of that coming up, but it's essentially the surface of the chloride cell in saltwater is concave. This is to facilitate accumulation of chloride concentrations such that they can actually passively diffuse into the water. Now, in the case of a freshwater adapted chloride cell, they have much more surface area. Rather than having a convex apical surface or a pit on their surface, they have many finger-like projections. The purpose of these finger-like projections is to increase surface area in the water to encourage greater opportunity and ability to uptake needed ions. Okay, so in terms of the ion transport model, how these things are actually functioning, they start with the very same ATP-dependent sodium-potassium pump. So you can see that on the right-hand side, the blood side of the figure. So once again, we are exporting 3 sodium ions into the blood in exchange for 2 potassium ions coming into the chloride cell. So what this is doing is this is helping to maintain a concentration gradient needed to facilitate diffusion of sodium into the cell. So any sodium that comes into the chloride cell from fresh water, it immediately is exported into the blood because we want to maintain very low concentrations of sodium in the chloride cell so that any sodium that's present on the freshwater side will passively diffuse in. And you can see that on the left-hand side, we also have a co-transporter here, and it is exchanging 1 sodium coming in for 1 hydrogen going out, or actually 1 ammonia going out as well. There's a couple of different ways it can function. Essentially, it's a cation exchange. And so we are importing sodium and we are exporting hydrogen through this passive transporter. Again, that's facilitated by maintaining low concentrations of sodium in the chloride cell at any time by actively transporting them into the blood. Okay. Now, the accumulation of CO2, carbon dioxide, in the chloride cell because of respiratory exchange, remember all the CO2 is going to be coming from the tissues into the gills and ultimately into the chloride cells. The accumulation of CO2 in the blood will diffuse into the chloride cell, and the accumulation of that CO2 is going to drive the bicarbonate buffer system to form additional bicarbonate ions and hydrogen ions. It's also going to shift the ammonia versus ammonium equilibrium to form more ammonia, or NH4. So increasing the concentration of those 2 materials, bicarbonate ions and ammonium ions, these will then diffuse out of the chloride cell down their concentration gradient into the fresh water in exchange for positive or negatively charged ions in exchange. So as the bicarbonate passively diffuses out, it's going to do so in exchange for a chloride ion that's brought in. Again, these co-transporters only really work if they are linked together. And so you can't just export a bicarbonate ion through this transporter unless you can import a chloride ion. So as bicarbonate ions accumulate in the chloride cell, they're going to drive that transporter to work more quickly and really bring in a lot of chloride, which of course is the purpose of this cell. It's the same thing on the cation or the positive ion side, in that the accumulation of hydrogen ions and NH4 is going to drive that cation transporter to export what is not needed by the cell in exchange for what is needed by the cell. So we're going to get rid of the garbage and we're going to bring in the sodium that the fish needs in exchange. It's really an elegant, efficient process. Okay. Now, there's also some thinking that the pavement cells may actually be engaged in some of this ion exchange as well. We know that there are active ATP-driven transporters in the pavement cells, which all those do is export hydrogen ions. So by exporting hydrogen ions, again, these are going to be accumulating in the tissues. They're going to be brought back to the gill by the circulatory system. They're going to passively diffuse into the pavement cell, and then the pavement cell is going to actively transport the hydrogen ions out into fresh water. It's thought that by actively exporting positive ions, you're creating essentially a separation of charge in the pavement cells, and this high level of negative charge essentially in the pavement cell may then attract or help to attract sodium ions that can then diffuse in. So you've seen on this slide and on the previous slide with the saltwater adapted chloride cells some indications of relative charges. So that's what the MV refers to here as millivolts. So in the chloride cells and in the supporting accessory and or pavement cells, because of all the ion transport that's going on, you can get separations of charges. You can get an accumulation of positive ions on one side of a membrane or an accumulation of negative ions on the other side, and this separation of charge may also facilitate attraction or export of these positively or negatively charged ions. So the electrical charge part of this is involved as well as the concentration gradients. And again, this is just another indicator of the same species that we talked about before, another description of sodium and chloride uptake by lamprey that have adjusted to fresh water. So lamprey are diadromous, so we showed the previous slide showing how they export sodium and chloride in saltwater conditions, and in this case how they import those same ions in freshwater conditions. I'm going to begin to talk now about the other major osmoregulatory organ in fishes, and that is the kidney. So the purpose of this diagram is to show you the basic morphology of and the basic structure of a nephron. So a nephron is the basic functional unit of the kidney. So the kidney itself is going to be comprised of many nephrons, but each of those nephrons is going to function largely independently to achieve filtration of the blood. So here you have a complete nephron, and the first component of it is shown on the left, and it's referred to as the renal corpuscle. This is the initial filtering unit, and this is the entryway into the nephron. So the renal corpuscle is actually comprised of two different items here. So it's the glomerulus, which is the capillary bed that's delivering blood for filtration. You can see that on the top here in red. And then the second component of the renal corpuscle is the Bowman's capsule. So this is the structure that collects the fluid from the blood into the glomerulus. So the renal corpuscle and the glomerulus, these were sort of referred to a bit earlier on when we talked about glomerular filtration rates. So glomerular filtration rates, which are adjusted for diadromous fish that are moving into and out of salt water, this is essentially the filtration rate is controlled by how much blood is delivered to the glomerulus. So if you want greater filtration rates, you're going to deliver more blood to the glomerulus, to this capillary bed for filtration. If you don't want to have as much filtration and you don't want to produce as much urine, you would restrict blood flow to that glomerulus. So the afferent arteriole there, the afferent referring to the arriving blood, might be slightly constricted to reduce flow to that glomerulus. Okay, and then you can see the efferent arteriole, which is the exiting blood leaving, but you can see it follows the rest of the nephron through before it leaves, and that will come into play here a bit later. So that's the renal corpuscle, the glomerulus, the capillary bed, plus the Bowman's capsule, the collecting area. From the Bowman's capsule, that leads into what's called the renal tubule, and it's comprised also of different components. So first there is the proximal tubule. You can see that listed here. This is the area of water resorption and reabsorption of organic solutes like glucose or amino acids. You don't want glucose or amino acids to be excreted in the urine. These are beneficial nutrients, and so those might be filtered out of the glomerulus into the Bowman's capsule, but those are going to be recaptured in the proximal tubule. From the proximal tubule, you lead into what's called the loop of Henle. This is where the filtrate, or the urine, is concentrated further for excretion, or is not so concentrated if your purpose is to try to produce high amounts of dilute urine. So the loop of Henle is comprised of two components. You have the descending arm and then the ascending arm. So the descending arm, or the descending limb of Henle, is permeable to water, but it is impermeable to salts. The longer the descending arm is, the more concentrated urine you can produce. Now, for terrestrial organisms, where water is a continuous limiting factor, the production of very concentrated urine is very important. This would also be true for marine teleosts that are not going to be producing a large amount of urine, and what urine they do produce, they want to be concentrated. So they can eliminate those excess salts if possible and not lose a lot of water. So for those species that want to produce a more concentrated urine, whether they're aquatic or terrestrial, they have longer descending arms or descending limbs in the loop of Henle, and you'll see why here on the next slide. So then you have the ascending arm, so the fluid is going to be moving into the ascending arm next. This arm is impermeable to water, and this is where you have active pumps that are pumping sodium ions out into the interstitium, so out of this vessel, out of this tube, into the area surrounding, into the tissue surrounding in the kidney. And the reason that this is done is to essentially drive water movement from the descending arm. And again, this will become a little bit clearer on the next slide when we can actually see the osmotic gradients. From the ascending arm, it leads into the distal convoluted tubule. This is lined with more active transporters that are there to reabsorb some of the needed divalent cations like calcium. This is also where you have exporters which can excrete phosphorus-based waste. So this is primarily focusing on divalent types of cations and their exchange. From there, from the distal convoluted tubule, it leads into the collecting tubule, which is essentially a connection to the bladder. And from there, of course, the urine, whether it's concentrated or dilute, is ultimately excreted into the environment. Okay, so now we're going to take a little bit closer look at the loop of Henle and the gradients that it creates in order to facilitate urine production, urine that is either very concentrated or very dilute. Okay, so you can see on the right-hand side the glomerulus shown in pink, so that's our capillary bed, and it's surrounded by that cup-like structure of the Bowman's capsule. So you have various types of osmolytes moving from the glomerulus into the Bowman's capsule, but they are not necessarily changing the overall concentration of solutes in that filtration. So you can see here the milliosmolality is, or the osmolality is 300 milliosmoles per kilogram in the glomerulus, in the blood, and it's the same in the Bowman's capsule. So this is likely a freshwater fish that's trying to produce fairly scant urine, or excuse me, very copious amounts of dilute urine. You can also note here that there is two different types of arrows. Red is for active transport, so those are the ones that utilize ATP, and then there's passive transport. So these are still transporters that only function in certain ways when they have certain ions present, but they do not require ATP to function. It would also include passive processes like water diffusion that doesn't necessarily need a transporter. Okay, so going down from the glomerulus into the proximal tubule and into the descending loop of Henle, you can see that there is an increase in the osmolality as you go down the loop of Henle. So we're going from 300 milliosmoles down to 400, 600, 900, and ultimately 1200, very concentrated, very high osmolalities at the base of the loop of Henle. The reason that you can see water is passively leaving the loop of Henle and causing this concentration of the filtrate is because of what's going on in the ascending limb. So in the ascending limb, you can see that there are many active transporters which are actively exporting sodium chloride from the filtrate, from the urine, into the interstitium. So they're creating very high concentrations of sodium chloride in the interstitium. Those high concentrations of sodium chloride then draw water out of the descending limb. Remember that the ascending limb is not permeable to water, so the only way that water can go into the interstitium and dilute that sodium chloride that's being pumped out is from the descending limb. So the descending limb diffuses water into the interstitium. Okay, so we create these very high concentrations. As we go back up the ascending limb, those concentrations decline as the sodium chloride is actively pumped back into the interstitium. Again, that's going to pull water out, but it's also going to facilitate recapturing of sodium chloride. If this is a freshwater fish, retaining sodium chloride is very important to them. So then you can see that the concentration of the filtrate continues to decline as more and more of that sodium chloride gets pumped out. And then ultimately it goes down or rather it goes up into the collecting tubules at at a concentration that's very low at 100 milliosmoles, ultimately leading into the collecting tubule and into the bladder. So it's being released at a concentration of 100 milliosmoles, much, much lower than the typical concentration of 300 milliosmoles per kilogram in the body fluids. So it's by this means that the fish is able to excrete excess water. Now, if a fish were in saltwater conditions and didn't want to eliminate excess water, wanted to retain water, their filtration rates and the concentration gradients that they're establishing through these active and passive transport processes would be adjusted appropriately. So there still are going to be anions, cations, and various electrolytes that they want to eliminate that are in excess. There are going to be those that they want to retain that are low in the environment and high in the body of the fish. So all fish are going to have a kidney that functions to a certain extent to export and retain certain ions, but the degree to which those processes will occur and the specific electrolytes that will be involved will vary based on whether we're talking about a freshwater fish or a saltwater fish. So again, just to drive that point home a little bit here, for marine teleosts, their renal apparatus, or their nephrons, they possess a glomerulus, they possess that neck segment, they have 2 or 3 proximal segments, and they do have a collecting tubule. So they have all the kind of basic components of the nephron. What's unique about marine teleosts is they have relatively small glomeruli, so they have very small capillary beds, and very few of them. Again, they really don't care about producing, about concentrating those ions and eliminating a lot of water. In fact, they want the opposite. So they tend to have a reduced blood filtration rate, and of course they produce scant urine. And they also are specially adapted in that their bladders are modified to absorb water one last time and to further concentrate ions for excretion. In other organisms, the bladder is less of an active organ and more of just a holding sac prior to urination. In the case of freshwater teleosts, they also have a glomerulus. They have a neck segment, 2 or 3 proximal segments. They have a very well-developed distal tubule that is water impermeable, which again facilitates the production of a high volume of quite dilute urine. And that's what we just discussed on the previous figure. So they have a large number of many well-developed glomeruli. They have high filtration rates. This is what allows them to produce high volumes of urine and eliminate all that excess water. Now, in their case, the bladder is a last-ditch opportunity not to absorb water, that's what they want to get rid of, but rather to reabsorb a few types of ions. So here again, we think of the bladder as being kind of just an inactive bag or sac that's holding urine prior to excretion or to urination, but the tissues forming the lining of the bladder do have some involvement with osmoregulation as well, either reabsorbing ions at the last moment in the case of freshwater teleosts or absorbing a last bit of water in the case of marine teleosts. With respect to elasmobranchs, so they have a glomerulus neck segment, 2 or 3 proximal segments, very similar approach to what many freshwater fish have. What's unique about their kidney and nephron system is retention of urea and TMAO. So they're retaining the urea to prevent the loss of salts to their environment and the loss of water to their environment. But of course, they're retaining that TMAO to prevent the protein destabilizing effects of urea from happening. Now, it says a high filtration rate and a high volume of urine. That's not entirely true in the sense that they do have a higher filtration rate than marine teleosts do, but it's not as high as freshwater fish. Keep in mind that like freshwater fish, marine elasmobranchs are they have a slightly higher concentration of osmolytes in their tissues relative to the environment. Remember, the typical osmolality for a marine osmoconformer is about 1,100 milliosmoles per kilogram relative to their environment. So they are going to be facing an influx of water, and so they do have a slightly higher filtration rate than marine teleosts to compensate for that. And of course, we've already discussed the rectal gland, which incorporates those cells that are very similar to those of the chloride cells in the gill, and that's going to compensate for that sodium and chloride influx that these fish will also face. So a little bit closer look here at what some of these cells look like. And also we'll use these figures as an opportunity to discuss physiological regulation of osmoregulation in fish. So here you can see chloride cells that are adapted either to seawater or to fresh water. So again, we have very different purposes and processes here. On the left, you can see the apical pit on the surface of seawater adapted chloride cells. Again, this is to facilitate the concentration of chloride ions to a very high concentration so that they will ultimately diffuse into the environment. You can also see the accessory cells and the pavement cells, abbreviated PVC. So the PVC, the pavement cells, have those tight junctions that don't allow ions to just work their way down through them. But in the case of the accessory cell, paracellular pathways, you can see that we have sodium passing down the pathway with relative ease. On the right-hand side, you can see not an apical pit, but you can see those finger-like projections on the surface of the chloride cell here. Again, those are serving to increase surface area so that any of the relatively rare ions that are present in fresh water can be effectively absorbed by this chloride cell. Now, generally speaking, the stem cells or the precursors to chloride cells can develop into either one of these options. So both seawater adapted and freshwater adapted chloride cells develop from the same stem cell, quote unquote, or the same precursor cell. Whether they end up becoming a seawater adapted cell exporting ions or whether they become a freshwater chloride cell importing ions is dependent upon hormonal signals that are provided by the endocrine system. So you can see here at the top we have cortisol and growth hormone, or IGF-1, insulin-like growth factor 1. Those two are responsible for inducing the development of seawater adapted chloride cells, whereas fish that require the development of freshwater chloride cells, they are typically under the influence of both prolactin and cortisol. So cortisol we commonly think of as being a stress hormone, but it has other functions in the body. Cortisol is considered to be hypoosmoregulatory, and it's commonly considered to be part of what prepares a fish for being in a hypertonic solution. In other words, cortisol is commonly expressed in salmonids that are migrating from freshwater into estuaries and ultimately into saltwater. As they go through this multiplication process, their cortisol levels are relatively high because the cortisol is responsible for preparing them for this transition to seawater. What cortisol actually does is it stimulates the activity of that sodium-potassium pump that drives both of these types of cells. So increasing cortisol will increase the activity of that ATP-driven sodium-potassium pump, which will increase the chloride export rate of a seawater adapted cell, but it will also increase the import rate of a freshwater adapted cell. So cortisol is kind of initiating, the process of ion export or import in both of these cells is initiated by the same pump. Cortisol increases the activity of that pump. So cortisol has this dual role here in terms of increasing the total activity of either type of chloride cell. Now, growth hormone, again, hypoosmoregulatory. It has similar effects as cortisol, and it would encourage the development of seawater adapted chloride cells. Now, prolactin, this is another hormone, is hyperosmoregulatory. It counteracts the effect of growth hormone and insulin-like growth factor 1, as well as cortisol during smoltification. So as a fish is transitioning from a freshwater osmoregulatory condition into a saltwater osmoregulatory condition, cortisol will increase the activity of both types of cells. Growth hormone is being expressed during this smoltification process, so that's going to upregulate and increase the number of seawater adapted cells, so that by the time the fish reaches the estuaries and the sea, they're prepared, they have plenty of those seawater adapted chloride cells. But also during that process, prolactin is being expressed at the same time to ensure that there are still plenty of freshwater adapted chloride cells to keep the fish functioning and osmoregulating normally up until the point that they do reach the estuary and they're ready to make that transition. So essentially at the end of the smoltification process, as those fish reach the estuaries and are ready to make the transition to osmoregulation in a seawater environment, the prolactin that's being produced is reduced, and ultimately they're not going to continue to produce prolactin. As a result, the freshwater chloride adapted cells are no longer going to be stimulated to function and proliferate, and will ultimately atrophy and become the cells will die and they will not be replaced. Of course, then the effects of growth hormone and cortisol will no longer be counteracted by prolactin, and so the seawater adapted chloride cells can then dominate. Okay, so we're just going to conclude by showing you some micrographs of what these cells actually look like in real life. So on the left-hand side in figures A and C, you see saltwater adapted mitochondrial rich cells or chloride cells. On the right-hand side in B and D, you see the freshwater adapted ones. So if we begin with the lower figures, C and D, you can see the very clearly defined apical pit of the saltwater adapted chloride cell. You can also see those pointed out by the arrows, these little holes essentially on the surface of the gill epithelium that represent those apical pits. Again, the purpose of the chloride cells in a saltwater environment is to encourage export of those excess ions. So they're not trying to do anything other than export those ions. If they had greater surface area, it wouldn't necessarily improve their ability to function. Remember that the export of chloride especially depends upon building of concentrations to very high levels, so that the levels of chloride in the cell are actually higher than the environment, and so they will passively diffuse down. So increasing surface area in a saltwater adapted cell will actually limit the ability of those chloride cells to, those chloride ions to become very highly concentrated and then ultimately diffuse into the water. So by limiting the exposure of the chloride cells to the water to just these little pits, it's minimizing the surface area. On the other hand, with respect to the freshwater adapted chloride cells, here greater surface area is an advantage. On the bottom you can see the finger-like projections that again serve to increase surface area of these chloride cells, and rather than having the cells represented by those small pits on the surface of the gill epithelium, you can see in figure B that the chloride cells here are essentially little patches where these finger-like projections are extending out into the surrounding water. So again, their morphology is reflecting their function. So that concludes the lecture on osmoregulation. As far as what I expect all of you to know in terms of the exams, I would certainly expect you to know the basic strategies of osmoconforming and osmoregulation, the relative advantages and disadvantages of those strategies. I would expect you to know the general processes that are involved, the challenges that are imposed for osmoregulators in a freshwater environment versus those in a saltwater environment. And I would also expect you to be able to describe or diagram the functioning of chloride cells in either a freshwater or a saltwater adapted environment. So there's a lot of information here, but those are the major take-home messages that I would expect you to know, especially the morphology and functioning of ion exchange in those saltwater or freshwater adapted chloride cells. That's it. Thanks.